Thank you for following me. Thank you for following. I'm still at church house, but no worried. Now, I was telling you that here we have considered a situation where, for example, I have carbohydrates. I've eaten carbohydrates, and after eating these carbohydrates have been broken down have been worked upon by digestive system into glucose. Glucose has been absorbed, transported by the blood to different parts of the body, where it is able to enter into the cells. After reaching into the cells, the glucose has been worked upon via glycolysis. It has undergone glycolysis and has produced pyruvate, and then pyruvate has moved from the cytoplasm, has come into the matrix of the mitochondria because oxygen is enough. And after reaching into the mitochondria, we have said now that the pyruvate has now undergone the cyclic reaction to form acetyl coenzyme A, which acetyl coenzyme A is now able to join the Krebs cycle, and then the process is able to continue to produce more ATP and so on. But I want to tell you that sometimes we don't only eat carbohydrates. Sometimes some of us, we eat some fats, foods which are fatty. Of course, though it is not healthy, but we are supposed to eat because we also need fats at some point for some reasons. Yeah, excess is what is bad. So what happens? What about when you eat foods which have fats? Is there a way how fats can also find themselves into producing of energy? The answer is yes. That when you have eaten fats, for example, when there are no carbohydrates, the fats that you have in your body can also be used to provide energy. How? When you have fats, for example, assume you have eaten fats, this is what will happen. When you have fats, we also call them lipids. We can also call them lipids. Lipids. Now, these lipids, we are saying they can be, when you eat them, these lipids undergo digestion, and when they undergo digestion, they will be broken down into fatty acids. And then they also be broken down into what we call glycerol, like that. So we have the lipids broken down into fatty acids and then glycerol. Now, in cases where, for example, you are fasting and you're not eating any other sugar and so on, now the fats that you have in your body are going to be happy, are going to also play an important role in enabling you get energy. How? We are saying those lipids, which are in form of fatty acids and glycerol, what will happen is these ones which are, for example, fatty acids, will undergo oxidation. Will undergo a series of reaction, and this series of reaction will involve oxidation. Actually, books call it beta oxidation. Other books call it beta oxidation. They will undergo oxidation, and in this oxidation, it means there will be loss of what? There will be loss of hydrogen. There will be loss of hydrogen atoms and so on and so forth. And through those series of reactions, a series of oxidation reactions, we shall have these fatty acids turn into acetyl-CoA. And when these fatty acids now turn into acetyl-CoA, now this acetyl-CoA will now come and enter into the Krebs cycle. And after it has gone to the Krebs cycle, as we are going to see, then in the Krebs cycle, we are going to have energy produced. You're going to have energy produced and other things, as we are going to see. So it means when you have fats, when you have fats, actually you don't have carbohydrates, and you don't have all these things, pyruvate and so on. The fats you have can also enable you to produce energy in this way. The fatty acids will undergo a series of reactions in your body, but basically the reactions are going to be more of oxidation, and then those fatty acids can be turned into acetyl-CoA. Then this acetyl-CoA now can come into the Krebs cycle, and this Krebs cycle, as we are going to see, we shall have energy given out in form of what? In form of ATP, and then the rest of the things will what? Will follow, as you are going to see. Likewise, when you have glycerol, here, what will happen? This glycerol, we are saying this glycerol will also undergo a series of reactions. And we shall have it being turned into pyruvate, for example. When you go into reading more, you find out that some of this glycerol at a given point, it first turns into, for example, DHAP. This DHAP, we talked about it under glycolysis. We saw when the fructose 1,6-bisphosphate was broken down, a reaction catalyzed by aldolase. We had two molecules being formed. One was pigar, and the other side was DHAP. So we are meaning if this glycerol can undergo a series of reaction and end up forming the DHAP, that means this DHAP can finally lead to formation of pyruvate, because we saw DHAP turned into pigar, and then pigar went into into pyruvate. So this means this glycerol can, through a series of reaction, can end up forming pyruvate, and then this pyruvate, when oxygen is present, this pyruvate comes into the matrix of the mitochondria, and then if it comes into the matrix of mitochondria, we are saying that it undergoes this link reaction, turns into acetyl coenzyme A, and after turning into acetyl coenzyme A, we are now saying this acetyl coenzyme A enters into the Krebs cycle, and then we have ATP produced more and more. Are we together? Yes. So that is the way. Now, we also, apart from lipids, the question is, teacher, is it only lipids that can give us, we have carbohydrates, we have got lipids, but we also have what we call proteins. We also have proteins. We also eat foods which are proteinous. Therefore, the question is, can proteins also give us energy? The answer is yes. Proteins can also give you energy depending on how, in some circumstances, especially when carbohydrates are not enough, or they are absent, or they are exhausted, or when protein are there. For example, assume you are fasting and you are fasting for 40 days and 40 nights and you're not eating anything. What happens is, yes, the stores of glucose will be first utilized. When they get done, we come to lipids. When the lipids are done, then the proteins, the body buildings, the food which you are ideally supposed to help you build the body will now start being broken down, and that's how somebody will start becoming thin and thin and thin and thin, until when they really become, they cannot become thin anymore and they end up dying. Yeah, so we are saying proteins can also be able to give us energy, but the question is how. In cases, for example, you have proteins. If I have, for example, proteins here, we are saying these proteins, for them, they are kind of different. Like they are not like exactly what we have seen for lipids. What happens is that these proteins are capable of undergoing deamination within, for example, within the liver cells. These proteins can undergo deamination. These proteins will first turn into, once they undergo digestion, these proteins can be turned into amino acids. Do you know the structure of amino acid? The amino acid has got a structure. For example, a structure which is like this. You have an amino group here, then you have something like this. You have a carboxyl group here. Remember I said amino acids are zeta ions. They form what we call zeta ions. And then here we can have an R group, and then here we can have an H. So that is the structure of an amino acid. And we said, for example, when this R is an H, that one becomes the simplest amino acid known as glycine. That one becomes glycine, the simplest amino acid called glycine. If this R is hydrogen, of course we know this R is usually an alkyl group, but assume it's not an alkyl group, it's just an H. That will be the simplest amino acid known as glycine. So we are saying these amino acids, what happens is, in case we don't have carbohydrates, we don't have lipids, and the only thing that we are remaining with is the proteins, which is in form of amino acids, what happens is that these amino acids will undergo deamination. And these amino acids will undergo deamination. They will undergo deamination. They will undergo deamination. And when they undergo deamination, of course, we are going to, in the process of deamination, we are going to remove the amino group. This deamination here, the word deamination, what does it mean? D is removal. Amination, amina is kind of is from amino. So in deamination, we remove an amino group. So this in deamination, during this deamination, we are saying the amino group is going to be removed. In this deamination, we are going to remove the amino group. This amino group the amino group is going to be removed, And once it is being removed, of course, it will go, this amino group will go with this hydrogen here, and it will go in form of what? It will go in form of ammonia. This amino group here will go in form of ammonia. So when this amino group goes in form of goes with hydrogen and in form of ammonia, we are saying now we shall remain with this other molecule here. We shall remain with this other skeleton here. We shall remain with this other residue here. So once the amino group is removed, we shall remain with the carbohydrates residue. You can call it the carbohydrate residue is what you're going to remain with. What are we trying to mean here? Carbohydrate, we are meaning this other group which is remaining here. From after moving this amino group in form of ammonia, this remaining thing. Of course, I've said this R is an alkyl group. Now we are saying this remaining thing can be able to join this remaining thing, this remaining group here, residue, can be able to join this system, in different ways at different points. Depending how this one will join this system will depend on, of course, what will determine how this residue is going to join the system will depend on the number of what? Will depend on the number of carbon, will depend on the number of carbon atoms which are present within this residue here. For example, if this residue is having 3 carbon atoms, for example, like assume this amino acid was like I think alanine. Assume it was like alanine. Alanine is an amino acid which has got 3 carbons. It is having 3 carbons. So if it is like alanine, assume this amino acid was alanine. After removing this, it means what is going to remain here is going to be having 3 carbons. Now, if it is alanine, now we are going to find that this alanine now will be able to come and can be able to be turned into pyruvate. Can be turned into pyruvate. It can be turned into pyruvate. You get? Can be turned into pyruvate. And then we are saying if this carbohydrate residue is having, for example, 4 carbon atoms, it is going to be turned into something that we shall have in the Krebs into the Krebs cycle. So when we now talk about the Krebs cycle, I think I'm going to re-echo again this pathway that happens, I will again repeat this one for proteins to show you how this carbohydrate residue can easily also join into. You get? Yeah, so if this thing has got 2 carbons, we can have it even come to acetyl coenzyme A. We can even have it join directly into where? We can even have it come and join into different areas here within the Krebs cycle, as we are going to see. So when I talk about the Krebs cycle in details, I think I'm also going to re-echo that so that we appreciate it. We appreciate how these proteins can also participate or come directly into this respiratory pathway to also give us energy in case carbohydrates are not there or lipids are not there. Is that one clear? So, our next step, we are now going to look at the Krebs cycle in details, and then we shall be able to join up to connect it further with this. So this is what this is what happens in the link reaction. So shall you join me and as we go into looking at the Krebs cycle in detail. Thank you very much. May God bless you.